Ladies and gentlemen, we now proceed to Symposium 8, a highly relevant session on pharmacogenomics, with insights into pharmacodynamic diversity, evidence, opportunities, and challenges in Sri Lanka, and lessons from global implementation. It is my pleasure to invite our chairpersons, Professor Chandanie Wanigatunge, President of the Sri Lanka Association of Clinical Pharmacology and Therapeutics, and Professor Priyadarshani Galappatthy.
Good afternoon, everyone, and welcome to Symposium 8 of the 58th Annual Academic Sessions of the Ceylon College of Physicians. I'm Chandanie Wanigatunge, and my co-chair is Professor Priyadarshani Galappatthy. So Symposium 8 is in keeping with our theme, Physicians for the Future, and we will be looking at pharmacogenomics, implications for clinical practice in Sri Lanka. And we have an exciting lineup of speakers who will share their expertise with us, and thereafter we will go for a panel discussion. So with that, I'll ask my co-chair to introduce the first speaker to you. Over to you, Priyadarshani.
Thank you, Chandanie. So I have the great pleasure of inviting the first speaker, who actually does not need any special introduction to this audience. He's Vindya Professor Vajira H.W. Dissanayake. He's an expert clinical geneticist, and he's also the Dean of the Faculty of Medicine, University of Colombo, as well as the President, Sri Lanka Medical Council. So over to you, Vajira, to do your presentation.
Thank you, Priyadarshani. So we are going to be futuristic, but there's no futuristic audience. So that's the shame of I guess we need to acknowledge that fact that, we may not be futuristic in the practice setting. So we got to change. I'm going to make a very simple case for why we should be implementing pharmacogenomics in Sri Lanka. And my colleagues who follow up, Prof. Priyanga and Dr. Vinod Skaria would definitely make the case in the clinic. So for anyone who is from abroad, this is where the Faculty of Medicine is. We have a historic wing, if you want to call that, as well as a modern wing in our faculty now. Just to begin with, there are these terms that are used, pharmacogenetics and pharmacogenomics, and those of us who are old enough would have heard the word pharmacogenetics when we were in medical schools, in my case in the 90s, some of you maybe in the 80s and so on. But the word genomics was coined later. And while the old generation talked about single gene, single genetic variant, single drug, we are looking at the genes, the entire genome, and the influence that they have on the metabolism of all drugs. If you take any drug, all of us know, once a patient takes it, the patient may benefit, may get cured, but may not have any toxic effects. On the other hand, the drug may be toxic with no beneficial effects. That's the two sides of the coin, as it were. Then there are drugs which will not be toxic as well as not beneficial. And then, of course, drugs which are toxic and not beneficial. But what's the scale of that problem, as it were? What opened the eyes of the world to the scale of the problem one of these was this article which was published in JAMA in April of 1998, which showed that there were more than 100,000 people dying of adverse responses to medication, and more than 2.2 million people experiencing serious reactions to medications, and many more failing to respond at all. This was in US. But at that time, in the 1990s, we had no way of predicting what that response was going to be. while that was the case in the clinical scenario, in the background, the Human Genome Project was in full steam. And the scientists were discovering more and more of these single nucleotide variants, which at that time we all thought would solve our problem because it would explain the variation in the metabolism of medications by individuals. The hype was so much that as at the turn of the century, in the year 2000, we had the launch of what was called the SNP consortium, Single Nucleotide Variants or Single Nucleotide Polymorphism Consortium, which was aimed at discovering all the variants in the genes. And if you look at this is a screen grab that I took as a small fellow in 2000 or 2001. You can see, if you look at who the partners in this consortium are, all the top pharma companies in the world, and the main research funder in the world, and the two biggest computer firms at that time, IBM and Motorola. The importance of pharmacogenomics, or the importance of, they all recognize the role that genetics and genomics is going to play in pharma in the years to come. Then, as time went by, FDA started taking notice of the evidence that was coming through. Then today, if you go to Google FDA pharmacogenomics biomarkers in drug labeling, you'll get this, and there are loads and loads of recommendations. And we had now the opportunity to use genetics and genomics to predict responses to drugs. That's what kind of brought about the change. It brought about era of what we are now calling personalized medicine. And if you look at the kind of the pharmaceutical industry, the pre-genomic era was, had the blockbuster model, whereas today we have the personalized model. The blockbuster model had narrow, clinical product portfolio of the companies. The personalized model will have a wide portfolio. If you look at the markets, they were producing a few drugs for masses. Whereas in the future, today and the future, we are looking at target therapies based on the genotype. The previous blockbuster model was aimed at the disease state, whereas now we are looking at the disease life cycle, because a drug that works in a particular stage of the life cycle may not be efficacious in the next stage, and so on. So you could see the change that moving from the old model to the new model was having, not only on the clinical practice, but also in the way the medicines were manufactured and will be manufactured in the future. Data such as this showing that in the blockbuster model, where a narrow range of drugs was being given to large numbers of patients, were actually not efficacious, was driving this change. And then also, if you look at the cancers, the realization that certain gatekeepers, as it were, in these, the pathways were key to whether a drug works or not. Also brought in this concept of precision medicine, and so on. And these concepts have now been incorporated not only to the guidance coming from regulatory agencies, but also in the guidance coming from those who are working on bringing pharmacogenomics into the clinical space. But what's the problem? All of us humans are not equal. we are different. We are genetically 99.9% equal. We are 0.1% different, but that 0.1% accounts for more than 3 million individual genetic level changes in between all of us. Although all of us here in this hall are 99.9% equal, we are 0.1% different, and that amounts to more than 3 million differences. So we wanted to look at the genetic profile or the diversity of the the variants in the pharmacogenomically important genes in the Sri Lankan population. We had a collection of samples. We collaborated with colleagues at the Genome Institute of Singapore. And about 10 years ago, almost now, we generated a set of data which is telling. And I'm going to take you through that data in just one slide. So these are genetic distance analysis in 4 square boxes, as it were. We were looking at these populations, the Sinhalese, Tamils, and the Moors in our country, Gujaratis in Houston, Punjabis in Lahore, Bengalis in Bengal, Sri Lankan Tamils in the UK, Indian Tamils in the UK, Indians in Singapore, the Chinese in Singapore, Malays in Singapore, and the white Western Europeans, the CEU. So the first box, as it were, shows the genetic distance of Sri Lankan populations, and there is hardly any distance. We are all clustering together. Then we go to the next, where we are comparing the Sri Lankan populations with all these Indian subcontinent populations that I mentioned. And you can see we are pretty much clustering together there as well. But the moment we go beyond, we compare the Sri Lankan populations with the Singaporeans, the Chinese and the Malays, the distance starts appearing. And then once you go and lump on it, the white Western Europeans, you see where they are. So the drugs that are the genetic variants in the genes that are responsible for absorption, metabolism, distribution, and excretion of drugs in our body, our variant profile is different to that of these other populations. And what's very important for us is, what are we doing? We are looking at evidence coming from the white Western European population, which is different. So we all look at the BNF and prescribe. So there's no wonder there's a lot of drugs that don't work for our population. We are like, using medicines which make the Western pharma rich and us poor, and poor health outcomes. So we got to change. So we need to look at bringing in the evidence and integrating pharmacogenomics into the medical sphere with evidence-driven decisions. So where is the evidence? One set of evidence I already presented, the case to show that we are different, and we need to acknowledge that and start recognizing that and working on it. And the next set of evidence is what my colleague will provide, Prof. Priyanga. We've been looking at the Sri Lankan genome. The past few years, we have sequenced more than 1,000, Sri Lankan whole exomes. We have data of our variant profile. And we've been looking at those variant profiles in various ways, looking at how we can bring pharmacogenomic-driven decisions to our decision-making process. So there have been about 7-8 papers that have come out that captures this evidence, and I'm sure Priyanga will talk about them. So, ladies and gentlemen, the time is right for us to capture the genetic diversity in our population into our decision-making process at all levels, starting from which drugs we should be bringing into this country in MRA level regulation making, as well as which drugs we should be using in our population, and also capturing, generating data on the dosages, generating data on adverse reactions, adverse events, and so on. So we can truly say that we are practicing evidence-based medicine in our country in a genomic era. Unless we do that, we'll be lagging behind. So with those words, I hope I have set the scene for the next two talks to come. And I'm particularly pleased that our colleague from India, Dr. Vinod Karia, is here today. Vinod was involved with us in sequencing the first Sri Lankan genome, and he's done a lot of interesting work in India. We look forward to listening to that. And of course, our colleague, Professor Priyanga Ranasinghe, has taken the data that we generate in the Center for Genetics and Genomics to a different level with the work that he has done analyzing that data from a pharmacogenomic angle. And we look forward to listening to both of them. Thank you very much.
Thank you very much, Professor Vajira Dissanayake, for that introductory remarks and for setting the stage for the next speaker. So it's my pleasure to introduce the second speaker, who actually needs no introduction. He was introduced very extensively in the morning when he delivered the PB Fernando oration. The PB Fernando oration this morning. But just let me introduce him again. Professor Priyanga Ranasinghe, Professor in Pharmacology attached to the Department of Pharmacology, Faculty of Medicine, is the first board-certified specialist in clinical pharmacology and therapeutics in Sri Lanka. His research interests are in clinical pharmacology, hypertension, and pharmacogenomics. He has published over 150 papers in peer-reviewed index journals and has an H-index of 53 with over 34,000 citations to date. He has been the recipient of over 30 awards for research, including consecutive presidential awards and CVCD Research Award. In addition, he has been ranked among the world's top 2% scientists in 2021, 2022, and 2023. So Professor Ranasinghe will speak to us on pharmacogenomics in Sri Lankans, evidence, opportunities, and challenges. Over to you, Priyanga.
Thank you very much, Madam, for the kind introduction. A very good evening to all of you. So, moving on from what Prof. Vajira Dissanayake said, I will be talking to you today about the evidence that we have generated, looking at it from a clinical perspective, taking some examples of common medicines that we use. Whether we like it or not, like AI, pharmacogenomics is also going to be something that will come to practice very soon, and something that we all need to be very familiar with because that is going to be the future. I'm not going to talk much about it. Professor Vajira talked about this, although we have patients with the same diagnosis. We know that they don't respond in the same manner. They respond differently. Some people do not respond. So in terms of efficacy, there is a difference. For example, studies have shown that about 90% drugs only work in 30 to 50% of the patients. And then there's the other side. When we start using medicines, people start getting adverse effects. And that is, again, a significant burden because, number one, patients may not be taking medicines, and then on the extreme end, they may be admitted with severe adverse reactions when you start using medicines. I'm not saying that pharmacogenomics is the only reason for these differences. There are many other reasons as we know of as clinicians, but increasingly we are recognizing that the genetic variants are responsible at least for some of these differences in terms of efficacy and also safety. We talk about the single nucleotide variants that are present in different populations and how these populations differ. Let me present to you some data about how common these variants are in different populations. So, I can tell you that everybody at 99.9%, most probably, in this audience, you will have at least one actionable pharmacogenomic variant in you. We may not be taking that drug, we may never take that drug during our lifetime, but that is how common these variants are, right? So it's important to recognize now, when somebody develops an adverse effects, okay, then it may be straightforward. You either reduce the dose or switch to a different drug. But imagine someone that doesn't respond to a medicine. Take, for example, an antidepressant, So due to a variant, they may not respond to that medicine, but then we use that medicine for some time before deciding that it's not efficacious. Whereas at the outset, if we had the information available to us that this particular medicine is not going to work on this patient because of the variant, we would have started on a different medicine from the beginning. And as I will show to you, there are common examples, even things like clopidogrel, where loss of efficacy will not be immediately evident. Adverse effects may be immediately evident when you start using the medicine with the doses, but loss of efficacy will not be something that will be immediately evident. If you don't respond to clopidogrel, you will come with another maybe a MI or a stroke when you for non-responders. So it's important to recognize that pharmacogenomics, at least, the loss of efficacy is something that may go on unrecognized for a long period of time and may have drastic consequences. I will take you through some examples. There will be lots of variants, so don't get lost in the variants, but I will highlight the clinical relevance as we go along. So these are some of the examples I will take you through. Some of the common medicines that we use: statins, warfarin, anti-cancer drugs, clopidogrel, for example. And I will highlight to you the difference in the Sri Lankan population, and as Prof. Vajira said, as South Asians, how different we are compared to other populations around the world and compared to populations from which we derive guidelines, which we derive dosages and recommendations. So ladies and gentlemen, let's look at statins. This is coming from one of our papers published here. If you're interested, you can go through this. But I will take a couple of, these are the variants that have been described, the genes that have been described, that are associated with statins, pharmacodynamics and pharmacokinetic differences. I will just take two examples and highlight to you with the mechanism that is involved, how we are different. Let's take the first one, SLCO1B1, which is a transporter that's encoded by this gene. That the particular variant, STAR5, with that RSID given, in our population, we found the minor allele frequency is about 18%. And the second one I'm going to talk to you about is it's a metabolizing a gene that encodes a metabolizing enzyme, CYP2C9, the STAR3 allele, which we found about 10% in our population. So what does this SLCO1B1*5 or the transporter does? It is involved in transporting statins into the liver. So that is the site of action. So if you have a mutation where this transporter is not working, then, or reduced function for that matter, or reduced expression, then obviously the transport into the liver, the site of action, will be reduced. Of course, with that, you may lose efficacy, but then the other problem is there will be when we give the standard doses, there will be a higher concentration in the plasma, which then means that the muscles are exposed to a higher concentration, increasing the likelihood of myotoxicity or muscle symptoms. Right? So this particular variant is a no function variant. Of course, we have 2 genes, so it may be reduced function at the end of the day, but if you have both together, you may have complete loss of function. So how do we, this is again the diagram illustrating the same. How does this 18% compare to others? So it's higher than what this variant is in East Asians, Americans, African Americans, although similar to the Europeans, significantly higher than in some of the other populations. What about the second variant, the CYP2C9*3? As I told you, it's an enzyme that metabolizes statins. Again, the variant is a no-function variant. So if you have 1 or 2 alleles, either you have reduced function of this metabolizing enzyme or you have no function. So again, the problem is there is reduced statin metabolism. So again, when you give a standard dose, there is less metabolism and inactivation, again increasing the likelihood of toxicity, and then again implying that maybe if we use lower doses, now some of the things that we hear, especially from clinicians, is that when our patients go up to the higher doses of, high-intensity statin therapy, they do not tolerate it. Maybe this is one of the reasons that they do not tolerate the higher doses. Maybe it's not required at the end of the day. So again, comparing, you can see the difference. It's about 10% in our population, whereas in other populations, it's much less. The other population data have been taken from online databases like PharmGKB, which collates published evidence. And you can see that in our population, it is much more. So the clinical implication being that maybe we require lower doses of statins, and one reason our people may be not tolerant to the higher end of the spectrum could be the presence of these variants in our populations. This is something we have to keep in mind. Let's move on to another example, another common drug still used in Sri Lanka, although gradually being replaced by the DOACs for various indications. Warfarin is still commonly used. So again, I will highlight to you a couple of examples and the mechanism involved in this. CYP2C9 gene and VKORC1 gene. These are some of the variants that we have identified in our populations. The RS, it's a single nucleotide variant ID. The RS stands for that, RS7294 of the VKORC1 gene, and the other RS variant in the CYP4F2. 40% in our population, both the nearly crossing 45%. So what does, this is the, I'm sure you are familiar with this, VKORC1 is the target for warfarin, and that's how it works, right? So what does this variant imply? It is an increased function variant. Increased function. So the activity of the enzyme is increased. So then to inhibit it, you will need a higher dose of warfarin. So how does that compare? You can see the presence is higher than other populations, especially when you compare against populations like East Asians. So which means when we go on with the standard doses, we might need to go several dose escalations before we reach the target dose, which means longer period of time that they may be not in target range of the INR. Then what does the CYP4F2 do? It is something that metabolizes vitamin K. So this is a decreased function mutation, which means vitamin K is not metabolized. So again, there is higher presence of vitamin K if you have this variant in higher numbers, higher percentage, which means again, you require a larger warfarin dose to gain activity. So you can see, ladies and gentlemen, this particular variant is quite significantly high in our population compared to all other populations. So is it right for us to then go by the dosing recommendations coming from other countries where our people, when we are starting with those doses, maybe for a longer period out of the target range? So with this in mind, there have been several initiatives where people have, if you go, there's something called warfarindosing.org website, where when you input the presence or absence of these variants, it will tell you what the dose that is required. So we are also in the process of developing with one of the PhD students a similar algorithm for our population, including the clinical variants and the GIS variants. So where you check the variants, and then with that you can get the ideal dose for the patient. So this is the clinical implication that I told you. One more example, taking this is again from one of our papers, which was very recently published. I will take XRCC1, which is relevant to platinum-based chemotherapeutics. There's another study that we are doing with Liverpool, UK of the DPYD variant. We have still not published that data. So this particular variant, the RS25487, is present 64% in our population. Now, this is an interesting variant, ladies and gentlemen. This particular variant is a decreased function variant. Now, this encodes for a protein, the XSRCC1 gene encodes for a protein that is involved in repair of damaged DNA, right? So, how does these platinum-based therapeutics work? They work by creating DNA damages, etc. So, when you have decreased function, that means the presence of these variants is actually good because they will respond to platinum-based chemotherapeutics more. But you can see in our population, the variant is less compared to others. So again, we may not respond. We may need higher doses, but if we use higher doses, they may get toxicity, etc. Right? Moving on from anti-cancer medicine to a very common one, clopidogrel. We all know that this is a prodrug which needs to be activated, and CYP2C19 is the gene that encodes the enzyme that activates this. So if you have reduced function or loss of function, you may continue to take clopidogrel without knowing that you are not responding to it until you get a stent thrombosis or a secondary attack. Right? So there are a lot of studies showing that this particular variant is highly common. I will show you our data comparing to others, so much so that in the UK, it is recommended that now it has made it to the NICE guidelines that you test for this for stroke indications. Now, even in the UK, as I will tell you, one of the challenges is getting the clinicians to take this up, the interest. So, although the stroke physicians and the neurologists has taken this on board, even in the UK, the cardiologists are still not on board, right? So, that is one of the challenges we face, getting people to understand this and get interested about this. So you can see, ladies and gentlemen, how the difference. I will show you our data, but you can see that when compared to the other populations, we are far away. The presence is much higher, right? So this is from our paper, which is very recently published, looking at this variant. 42%. How does it compare? A drastic difference. A drastic difference compared to Europeans, Americans, even East Asians, especially the European and American populations, from which we derive guidelines and etc. I know there is one study or clinical trial going in this country where we are trying to see, okay, let's forget about the variant, but can we give a higher dose? And whether people, rather than giving 75, can we give 150? Yes, it may work if you have decreased function, but if you have two mutations where there is no function, even if you give the higher dose, it's not going to make a difference. So as I said, ladies and gentlemen, there are many other examples. I'm not going to take you through all of this, but we have published these in various papers. Especially take the example of the antidepressants and clopidogrel, which I told you about. As I highlighted at the very beginning, if you get an adverse effect, yes, that's fine. You change the medicine or reduce the dose. But if you don't respond, you go on for years and years, maybe months, without knowing that from the beginning, you are a non-responder, which is something to keep in mind. So these are some of our other papers that we have published with regards to HIV medicines, the anti-infectives, with regards to immunomodulators and biologics, the different variants that are present in our populations. I'm not going to take you through all of these. We recently published a systematic review also looking at all pharmacogenomic studies that have been done in Sri Lanka thus far. Not many, but I think that is an important value addition to look at all these different studies, not only by our group, but other studies that have been done in this area. But ladies and gentlemen, not only in Sri Lanka, it's been a challenge implementing pharmacogenomics around the world. This interesting paper looked at the different success stories with regards to pharmacogenomic implementations around the world. And a large majority of these programs are still in high-income countries, of which a large portion, I suppose, supported by how the US health system is arranged and governed, is in the USA. So even in other developed countries like the UK, yes, it is gradually making its way into clinical practice, but it has been slow. What are the reasons for these differences, and how do we move forward? So the first step obviously is to understand what is relevant for our population. So as you can see, and as Prof. Vajira also highlighted, we are different. We are similar as South Asians, but then we are further away from East Asians than other populations. So like the work that we have done, it's important for us to characterize and appreciate the variants that are there in our population. That will be the starting point. What do we need to test for? There is no point in testing for a variant that is there in a European population if that is not found in ours. There are so many different variants. So we need to characterize, and some of our work is actually to enable that, to characterize the different variants in our population. Then we need to, okay, fine, we have identified the variants, but people may ask, you have looked at cohorts of people who are not taking the medicine, what is the clinical correlation and so on. So you need to then think about, because as I told you, genomics or genetics is not the only reason for differences. So we need to do clinical correlational studies. We need to increase awareness, which is what we are doing during these different symposia. Unfortunately, although not very well attended, we need to raise awareness about, especially among the clinicians who are at the end of the day going to be the end users of this. And as I said at the very beginning, whether we like it or not, this is going to come in the future, and it is important that the clinicians are aware about it and educate and train. Of course, access to test and having cost-effectiveness test is also a challenge, but over the years, not only the access, but things have become cheaper, and it will continue to become cheaper. So that will gradually improve. So in summary, ladies and gentlemen, as you saw, genetics are being increasingly recognized as an important factor affecting variations in how people respond, not only in relation to adverse effect, but also in terms of efficacy. And like how we use renal functions, like how we use liver function, like how we use clinical data to customize the dosages or medicines to patients, in the future, pharmacogenomic data is also going to be a utility, an investigation that will be available to clinicians to target or decide on the medicine and to decide on the appropriate dose for your patients. Implementation is slow, but I think we are getting there gradually. So with that in mind, I would like to end my talk. Thank you very much.
Thank you very much, Priyanga, for that excellent talk, highlighting the Sri Lankan data coming from Professor Vajira Dissanayake's genetic laboratory, the many genetic variations that are experienced, that are noted in our population. I'm sure that will have a lot of clinical relevance. We'll go into that during the discussion. It's now my pleasure to invite Dr. Vinod Skeria and to inform about him. Dr. Vinod Skeria is a clinician scientist and researcher specializing in precision medicine, medical genomics, and computational biology. He has played a pioneering role in advancing clinical genomics in India, contributing to genome sequencing, rare genetic disorders, pharmacogenomics, and infectious diseases. As a co-founder of key national genome initiatives, he has established infrastructure and frameworks that bridge clinical practice and genomic science, enabling large-scale patient impact. He has over a decade of experience with numerous high-impact publications, and so he is going to talk to us on pharmacogenomics in clinical practice, lessons from implementation. So I think it will be useful for us to learn how India has implemented some of the genomic data into clinical practice. Over to you, Dr. Vinod Skeria.
Thanks a lot for the nice introduction, and thanks Professor Vajira for inviting me here, and the organizers for inviting me here. Well, I have a very difficult talk to present, lessons from implementation. Well, nowhere on earth has pharmacogenomics been implemented as it should have been. So I decided to rather talk about how it has not been implemented, or what are the mistakes that I've done in my life. Well, it's always a pleasure to be in Sri Lanka, quite long association of around a decade and a half. And I think it is probably the right time to also celebrate a decade and a half of the Sri Lankan genome. Professor Vajira is in here. Now, going back to genomics and pharmacogenomics, and I'm sure you have all seen this particular slide quite very often used across multiple presentations. We all know that not all drugs given to patients work in the way they were supposed to work in the first place. And that's largely because of the fact that in almost all cases, clinical trials are done on groups of individuals. And what we essentially try to do is to average observations across the groups. So there is practically no single individual in the clinical trial which will represent the group. And therefore, there's always an average patient that represents nobody. Now, how do we really look at this in a genomic perspective? And my life in pharmacogenomics and genomics starts with the first Indian genome, the 7th genome to be done, and probably the first genome to also have a very dedicated section on pharmacogenomics. And the idea was very simple at that point in time. Can we really put all the evidence at that point in time to decide how an individual should be given medications by his or her doctor? And that ended up in this small paragraph out there. We extended that further into looking at multiple more drugs and also building in one of what we call as a Google Maps for pharmacogenomics. essentially taking every single drug by classification through the transporters, the metabolizing enzymes, and the targets to see if there are mutations out there, can it affect the pathway of the drug. Just like you do Google Maps from point A to point B, if there's a roadblock out there, it will tell what needs to be done for every single drug at any single point of time, provided you have a whole genome sequence. Now, of course, 1 genome or 2 genomes or 10 genomes are not going to make a difference. So we decided to essentially look at a larger cohort of individuals. These were arrays at that point in time, sometime in 2010, 2011, with my colleague, Dr. Dwaipa and Bharadwaj. We essentially tried looking at pharmacogenomics of clopidogrel. And what we essentially showed is that the Indian population frequency for the pharmacogenomic relevant variants are quite distinctly different from the global population. I don't need to put in a p-value in there. The size of the dots are representative for themselves. What it essentially means that we have a much better intestinal absorption, much better bioactivation, and much better inhibition of platelet aggregation on an average scale. Now, of course, when you look at this on a population scale now that there were many more population genomes available at that point in time, we found this very, very interesting observation. So what I need you to do is to essentially follow from left to right and just focus on the red part of the pie. The red part of the pie is what we call as a poor metabolizer for clopidogrel. And what you see here, as you move from west to east, the part of that pie increases in proportion, which is practically non-existent in the Western world, but now very high frequency, almost 12-13% in the Eastern world. Now, what does this mean? This means that probably we are giving clopidogrel the wrong dosage to the wrong people. It's largely because of the fact that a poor metabolizer will largely be resistant to clopidogrel because they have STAR2, which is a homozygous mutation. Clopidogrel is a prodrug, it will not be metabolized, and therefore there's practically nothing to inhibit aggregation. Now, this was really, really interesting to look at, but what we really see is macro scale observations. Now, what we wanted to also understand is, are these macro scale observations really consistent at a micro level? Now, at that point in time, we are fortunately looking at the Qatar population genomes. Qatar is a very, very small country with approximately 150,000 natives, largely stratified because of marriage practices and consanguinity. And what we show here is, while on a macro scale you could have allele frequencies, on a micro scale, depending upon what tribe or community you belong to, you would have very distinctly different allele frequencies. Now, that necessarily means that while we can derive insights on a macro scale, what we need to really look at is in a micro scale at a level of population, subpopulation, community, or tribe. And this is very more relevant in the Eastern world and Southeast Asian world, given that we have practices which preclude outbreeding of individuals. It could be cultural, it could be religious, it could be geographic, but we do have inbreeding in multiple different forms, what we call as consanguinity or endogamy, widely practiced in the population. And this could really, really change the allele frequencies of clinically actionable genetic variations at a micro level. Now, of course, now take this to a population scale in India. I'm not sure you would appreciate how big India is. 1/6 of every human on earth is an Indian. But of course, we are not a homogeneous population. We have 4,600 communities which are endogamous or consanguineous in nature. So that means that you need to extrapolate Qatar into 4,600 communities, and you will quickly realize where the complexity of the problem is. Now, of course, when you look at communities, you will also have population frequencies which are very, very drastically off the chart for subpopulations. And this is one example, just one example of the many that we have now identified, founder mutations of pharmacogenomic importance in small communities. And this is a small community called the Chettiar in southern India. And a paper published essentially suggesting that 1 in 24 individuals in this particular population would be susceptible to succinylcholine apnea because of homozygous mutation in the BCHE gene. Now, until yesterday, I thought, okay, this is just a South Indian problem. While walking through Gall Road, I realized that there is a shop out there which says some Chettiar jeweler. So that's also your problem, not just my problem. And in fact, what we show is that approximately 1% of South Asian populations do have these genetic variations. Fortunately or unfortunately, we don't use succinylcholine anymore. But if you have ever talked to a member of the community, you would realize that every one of them would remember at least one close blood relative who has been lost to surgery. Now that opens up a very interesting question on what is really, really prevalent, right? And of course, there are macro scale differences, there are also micro scale differences, but in a population perspective, we want to understand what is really prevalent. And the only way to me that you can estimate prevalence is to be able to sequence apparently normal people. And of course, to do apparently normal people sequencing, you need to have a population genomics initiative. In 2019, India initiated on this path with the Indigen program, which we co-founded with my colleague, Dr. Sridhar. And the idea was to sequence approximately 1,000 odd genomes from across cosmopolitan populations in India, not tribal groups, not consanguineous communities, but cosmopolitan populations across the country, to understand what is really prevalent, not just about genetic diseases, but also about pharmacogenomics. And of course, 1,000 in 1.4 billion is a very, very small number. So you would realize that if you find an allele frequency difference very large enough, this would impact millions of people within the country. Now, of course, there are obvious candidates that come to the top list. This is statin. My former speaker indeed mentioned about statins, about the transporters as well as the metabolizing enzymes. What I sort of suggest is that approximately one third of individuals would require a dosage modification for at least one of the statins currently available, and approximately 10% of individuals would require a reduced dosage for most statins. So in other words, the dosage probably that we are giving for statins is probably on the higher side. We can actually live with a much lower dosage, provided we can actually show also the clinical outcomes are very similar in such a case. And these are essentially going to drive the next generation of studies. Now, of course, if you do a whole human genome, and if you do it for 1,000 odd individuals, what we also realized is something that opened all of us eyes. On average, every Indian would have 8 clinically actionable pharmacogenomic variants as of today's evidence. 8 clinically actionable pharmacogenomic variants as of today's evidence. And some of them are quite very consistent, like for example, 2C19, poor metabolizer, STAR2, homozygous, is approximately in 13% of individuals. It affects a variety of drugs, including, for example, clopidogrel, making it a poor metabolizer for clopidogrel. So in other words, these 13% of individuals would probably require a second level of drug for example, ticagrelor. And therefore, genetic testing might really be important. And of course, today it is already in the guidelines. We don't need to reinvent the guidelines to be able to tell clinicians what to do. But unfortunately, clinicians are pretty much not interested. Cardiologists specifically are not interested because what is really important for them is the stent in the heart. What happens after that is somebody else's problem. Now, having said that, can we really now use this information to do 2 things? One, of course, can we impact individuals? And second, aggregated information, can it derive the cost reduction of testing to make very cost-effective, affordable, accessible solution for testing? The hypothesis is very simple. For people who can afford to get a whole genome sequence, they can anyway get a whole genome sequence. The guidelines today on how to interpret the data, you could actually provide them information about whether you should get for any given drug in the guideline a normal, reduced, increased, or an alternate drug because you have an adverse drug reaction. But you can now aggregate it at a population level and ask a question, what is really prevalent, and try to make the prevalent genetic variations into a test which makes it affordable. I'll give you just one example of the many. This is for 5-fluorouracil, DPYD. This previous speaker has sort of alluded to it. 5-fluorouracil is metabolized by a single enzyme, DPYD, which metabolizes approximately 80% of the drug and makes it inactive. It's a very commonly used anti-cancer drug. So the idea is that there is already observations in the population that approximately 10% of individuals would have pharmacogenomic variations. Nobody had any data at this point in time. So we went back to the population database. We looked at what is prevalent in the population. We arrived at 15 genetic variations, irrespective of the evidence which is prevalent in the population. We quickly went to a clinical cohort working with Dr. Pavitran from the Ahmedabad Institute of Medical Sciences to sort of validate these 15 variants. And what we suggest is that just there are 4 genetic variations which account for 90% of all clinical pharmacogenic actionability. So what we can do is essentially put these 4 genetic variations into a multiplex RT-PCR test. which is now offered as a program for many years and currently available in the diagnostic market from Dr. Lalpath Labs. So this makes it very affordable, accessible to people because you can do it at a few hundred Indian rupees. You can do it much faster because it's an RT-PCR test, not an NGS test or an array test, and therefore can be scaled quite very better. Now, having said that, I'll also allude to the fact that sequencing is becoming cheaper, and we are probably at the best times in the history of genomic sequencing that it is moving from discovery into clinical applications, and probably the next 1 decade will see significant cost reductions and improvements in throughput, which would also make it one of the routine clinical workups that are currently being used. The advantage of a whole genome being that it covers practically all kinds of genetic variations. And today when we talk about pharmacogenomics, we're largely covering the single nucleotide variations. We typically ignore the copy number changes and deletions, which are also not very rare. Having looked at enough number of genomes, these are quite significant variations or percentage of genetic variations that we currently miss. But a whole genome covers practically every single kinds of genetic variation that we know today. Now, of course, we have dozens, if not hundreds of individuals who are accessing whole genome sequencing as part of their genetic testing. Can we really provide a pharmacogenomic output to that? The answer is probably yes. So a couple of years ago, we launched India's first whole genome sequencing initiative, where we could provide information on a variety of facts, including clinically actionable pharmacogenomics, based on the current evidence at this point in time, along with clinically actionable findings, incidental findings, HLA, cancer predisposing syndromes, etc., etc. For people who can afford to take, this makes a very wonderful opportunity. And today we report 98 unique drugs and around 29 genes for which there is a level 1A or level 1B evidence for actionability in pharmacogenomics, and also conforms to the CPIC guidelines. Now, having said that, the most important component here becomes how do you really communicate this information back to the patient? And this is something that is sort of underrated or under-discussed in practically all sessions in pharmacogenomics, but probably the most important part of pharmacogenomics. Of course, you can do sequencing, you can do the test, but what is the patient going to get out of it, or what is the patient going to understand out of it is really, really more important. And here you need to really strike a balance between promoting the clinical impact versus minimizing the adverse psychological behavioral problems. And even for pharmacogenomics, I'm sure if you have done a counseling for pharmacogenomics, people are going to ask, does that mean that I have a disease? No, you don't have a disease. You're perfectly fine until you get the drug. And you also need to have very standardized ways of describing this. And of course, CPIC today has a guideline on what are the terminologies that are required to sort of put in a functional descriptor or a phenotype descriptor, so that there is no change between two individuals who is communicating the same information. For example, you cannot really tell a patient that you have a mutation for so-and-so drug. It does not really make a sense because the person is perfectly normal. Now, the other way is to essentially communicate this at point of care. And for that, you need to have electronic tools. And this was launched in 2019 by the Honorable Minister of Science and Technology at a point in time as part of the Indigen program. So every participant of the Indigen program actually has access to their genomic data, for the clinical information on the genomic data, pharmacogenomics being prime. So the idea is to essentially provide this information in a secure, privacy protected, scalable, point of care and clinician enabled methodology, so that the clinician can act upon that information without having to order a test. Now, having said that, the only thing that we need to realize after working many years in pharmacogenomics is probably we need to stop talking to doctors. We should start talking to computers. Because as electronic medical healthcare or ecosystems are growing, the clinical notes, their investigations, their diagnostic outputs, including genomic data currently, and we need to have an ecosystem where patients and clinicians can talk to computers rather than probably talking to each other. And the other important component, which is often understated, is the education of not just clinicians, but also the public on how this is really, really going to change not just their lives, but also the lives of their patients. And I was fortunate to have run quite a number of education programs. It's quite significant participation from Sri Lanka, not just about pharmacogenomics for clinicians, but also for the lay people. Now, having said that, the world is probably going to change as we speak. I'm not sure you've noticed this news article which came in probably a month ago, with the UK NHS program to sequence practically every single child born into the NHS system over the next 10 years. Now, that would necessarily mean that every one of the children would have genetic information, including the pharmacogenomic information, preemptively addressed at any point of time. And clinicians don't require to order the test anymore. They just probably need to go back to the data and ask the question, what is the dosage that I need to give for this particular individual? I think that is going to be revolutionary, and that's going to catch up not just in UK, but probably across the world as the cost of sequencing comes down. Now, having said that, this is an old saying that superior doctors prevent the disease, mediocre doctors treat the disease, inferior doctors treat the full-blown disease. Probably, should patients be waiting for the superior doctors, or probably they would take it upon themselves to do their whole genomes? Probably time will tell. Thank you very much.
Dr. Vinod Karya, for that very interesting presentation. May I now request the three speakers to come on stage, and also the three panelists who are to join us to come on stage. The three panelists are Professor Jennifer Martin, Senior Physician, Professor of Health Policy, Board Director, and President of the Royal Australasian College of Physicians. Dr. Dineshani Hettiarachchi, Specialist in Medical Genetics, Senior Lecturer in Anatomy and Family Physician, Department of Anatomy, Genetics and Biomedical Informatics, Faculty of Medicine, University of Colombo, and Dr. Vindya Subasinghe, Consultant Clinical Geneticist, Lade Ridgeway Hospital for Children in Colombo. So I'll hand over to my co-chair, Professor Priyadarshani Galappatthy, to start the panel discussion.
Thank you, Chandanie. So I think we had three very interesting talks, which actually highlighted the scale of the problem pertaining to genetic variations in populations, particularly in Sri Lanka, as well as in India and some of the other populations. So in starting the discussion, maybe I would like to ask Professor Vajira Dissanayake, and based on what Priyanga highlighted, particularly on clopidogrel, where 43% do not have the activating gene, whether we should be promoting testing for CYP2C9 in our populations before they are started on clopidogrel, because I think that's a very important clinically relevant problem. And Priyanga highlighted that, rather than adverse effects, which is quite obvious, lack of efficacy will not be obvious until they develop a MI or a stroke. So I think you have implemented testing in the private sector as well. And so is it available, Vajira, freely for anybody if they want to do, either in our genetics department or in the private sector, or what do you suggest?
Yes, so I think I can tell you, as you know, the first drug where the FDA mandated genetic testing was for warfarin. I'm correct, Vinod. It was 2008, and that was, at that time itself, we introduced the tests. But the challenge is the doctors not taking it up, right? So, like that, there was a whole series of tests, including for clopidogrel, which we introduced, but doctors were not taking it up, and neither were we kind of pushing it that way. But that is where the evidence is required, and so we did not really have population level data that we could take to meetings like this and showcase and start a discussion on. And I think that is where we are at the moment. So we have got the evidence, and we need to now start the discussion with groups of doctors. Here is the evidence. These are the benefits that genetic testing will bring to your patients, and therefore let's see how we can bring these tests to the patients, because genetic testing is the cost of testing is volume driven. If you have more tests, the cost will come down. If you have few tests, the cost will be high, and so that is where we are. So some of the tests that were showcased today have been implemented both in the faculty as well as in the private sector. But As I said, we have really not pushed it to the doctors. It has been just word of mouth, few doctors who want to use it. But now I think we have the evidence and we have the guidelines coming in both from the FDA as well as the clinical pharmacogenomic implementation guidelines. So now it's the time for us to start integrating this into clinical practice. And so I had a chat with Prof. Prasad Katunanda and said, let's initiate it at the College of Physicians session, and that's why we are here today.
Thank you, Vajira. I think it's a very, very important, factor that you have, highlighted, particularly what are relevant for our population. I think, somebody would spend about maybe, closer to $1 million in the private sector to have a stent. And then in the government sector also, it will be government is spending maybe even half of that. And then, whatever the cost that takes, it may be even a few thousand rupees for a genetic test rather than having that stent thrombosed. I think it is really worth it. So I think that is something that really probably taking it to the next level implementation. So I think that's a very, very important thing. Probably I think you will take it up with the cardiologists, and I think they will be probably favorable.
Think about, a patient on antidepressant who does, who's taking a drug which doesn't work.
Exactly.
Like we know of potential, people like that. And so we really need to take cognizance of all this data that's now being generated. And it's always the question has been, genetics is something Western, is it relevant to us? Now we have shown it's relevant to us. And the work that we know that team has done in India, they are by far, I think, the foremost pharmacogenomics group in India. And also now what Priyanga is doing, dissecting our data from all angles is going to really make us, make it possible for us to have that conversation on implementation.
Yeah, okay, thank you. I would like to now turn towards the other two panelists. I know Dineshani Hettiarachchi. Dineshani, you have been doing a lot of work regarding rare genetic disorders. So do you see what is the relevance of pharmacogenomics in the rare genetic disorders as a scenario?
Thank you, Madam. So as the speakers before in the symposium mentioned, the main goal of pharmacogenomics was to look at the drug efficacy, optimize dosing, and to avoid side effects. But in terms of rare diseases, I think most of the rare diseases were deemed incurable before 40 years. So after the Orphan Drug Act came into play, which was in 1983, I believe, FDA gave some incentives for some pharmaceutical companies to develop orphan drugs that is specific for rare diseases. With that, there has been tremendous development in this sphere. And now, as most rare diseases are monogenic conditions, that is, they're single gene disorders, when they know the molecular problem, that is the variants, the molecular targets are clear, it's easier to develop the drugs. So now we see for cystic fibrosis, Duchenne's muscular dystrophy, even certain motor neuron diseases, even hemophilia, Fabry disease, and so on. There are a lot of treatment that is coming into the forefront, but the problem is the cost of these treatments. So if, especially with rare diseases, since the population affected is very low, the cost the pharmaceutical companies have to invest in creating these drugs are very high. So if this cost comes down, then we can definitely find more patients to treat, and the availability of the drugs will be more. So I think since of late, there's a tremendous sort of interest in finding curable treatments for rare diseases.
Okay, thank you. And moving on to Dr. Vindya Subasinghe, you work as a clinical geneticist in the LRH, the Premier Children's Hospital. So what do you see as the implementation of some of the data that was shown here and the relevance into pediatric clinical practice of pharmacogenomics?
Thank you, Madam. Of course, yes. So in pediatric practice, pharmacogenetics in general, as we discussed, we need to think of the responders, non-responders, differential responders, and adverse effects. So we talked about the mainly in adult care treatment regarding the differential respondents and non-responders so far. But in terms of pediatrics, we are mainly having problems with the dire things. I actually, Dr. Dineshani mentioned is the personalized treatment is certain rare diseases like cystic fibrosis. So we think that these conditions are rare, but they are not that rare. It is in a country like ours, I think it is mainly underdiagnosed. Even cystic fibrosis, now that we have special treatment Evacaptor for certain genotypes, it's really important that we diagnose our patients and know their genotypes. So that's one thing. And on the other hand, if we talk about this cancer treatments, we need to think of the adverse reactions. So in that case, actually currently we don't readily practice doing pharmacogenetics in cancer patients, pediatrics, mainly leukemias. But I've had some discussions with the pediatric oncologists to get some their insights. I think they have taken a step forward to press the government to do certain genetic testing in DPYD and TPM. Those type of things, because it's really important, rather than otherwise we are not doing justice for the patients. So those are the main things, two things. And in addition to that, in clinical practice, now that I've started working as a geneticist very recently, but the more workload that I'm dealing is from neurology. So I'm doing a neurogenetics clinic together with a pediatric neurologist. So the other main thing that we encounter now and nowadays diagnose more, I think, are the epilepsies, the refractory epilepsies going into mostly due to genetic epilepsies. So in that case, what I notice is it's the usual practice of managing epilepsy patient is to start with one drug, at the second one, and depending on the response, at the third drug. And when it is not responding, you think of a genetic epilepsy, and then do you do genetic testing? And we have done so many genetic epilepsies with Professor Vajira, Dissanayake, sequence them to find the etiology of the epilepsies, and then to find that we have given a contraindicated drugs for them already. So, and we have having the genetic diagnosis with the whole exome sequencing, we have transformed their lives. Actually, there are a few examples I have encountered very recently. So I think it is the high time that we look into this matter and we do justice to our patients with the knowledge of pharmacogenetics.
Thank you very much. I think that's quite interesting and relevant how this information is translated to clinical practice and improving the lives of patients. So you want to add anything about the Indian experience in implementation in India? Because I think our population and Indian population are also fairly similar and in genetic makeup as well, as we saw. So to what extent do you use, say for example, in using this in the clinical practice?
So that depends upon the center and that depends upon the clinician, but generally the adoption or the use of pharmacogenomics is on the lower side, no doubt about it, and that is true globally. Now, to me, I think the reason of clinicians not adopting pharmacogenomics has nothing to do with the evidence. The evidence is there, it's already in the guidelines. Nothing to do with the cost. The cost is already affordable compared to the errors that you can make on the therapy. And what the patient already pays for that error. So it's not about cost, it's not about evidence. I think it's largely because of the mindset. And that mindset, to me, largely stems from the way we teach clinical medicine, right? Because, being trained as a clinician, a clinical journey starts with a patient. Right? A patient starts by definition with either a symptom or a sign. Clinicians are not taught to act upon individuals who don't have either of it. So it is just ingrained in our brain that don't do something until you find something. Right? And in my experience, when we launched the DPYD test, it was offered free of cost. Expectation is that all cancer patients who are taking DPYD would access it because it's free. It's covered in the program, so it should be free. A retrospective audit of the data shows that 95% of the requests are from clinicians who already have a patient who has an adverse event. Essentially, they're trying to prove to themselves that I didn't make the mistake, it was a genetics error. Right? It's just the way we are taught how to act in clinical medicine. We're not taught to act upon something that we don't see. So I think that's what we need to change.
About the DPYD testing, with something I didn't share. Something that we found from one of our ongoing research projects. At the moment, we are doing a study with Liverpool to characterize the rare variants of DPYD gene, where we are recruiting patients who have developed the severe grade 3 or grade 4 toxicity to DPYD, the medifluorescent drugs. Retrospectively from the cancer hospital in Mahararama, and we have got involved all the clinicians to identify patients. There's one clinician among all who preemptively does DPYD testing in the private sector. It's available in one of the private hospitals. It's about 25,000. So 2 of the variants are checked. And if the variant is present, he doesn't give the medicine. He uses the alternative. So we have recruited 25 patients so far. Only 1 patient is from that unit. All other 24 patients are from other units. So that is a success story. Clinical initiation.
Okay. Right. Thank you, Priyanga. Yes, I think I would like to now request Professor Chandanie Wanigatunge to wind up the session. We are, on time.
Yes, so thank you very much to the speakers and the panelists for a very interesting discussion. And I think it is something that we need to take forwards because it will improve the life of the patient as well as reduce a lot of problems to them. So probably we need to take this discussion beyond this point. Although the attendance was poor, maybe something we need to work on. I don't know why it was poor, but let's see what we can do from the college and the other associations. So we have our certificates of appreciations from the College of Physicians to the 3 speakers and the panelists. So I'll invite my co-chair, Professor Priyadarshani Galappatthy, to hand over the certificates to Professor Vajira Dissanayake. And to Dr. Vinod Skaria. Priyadarshani, you have.